Hello, everyone, and welcome to this edition of the Permit COE webinar series, where Joaquín Dopazo will present digital twins for drug repurposing by integrating mathematical modeling and machine learning, bridging the gap between in silico and real-world data. My name is Marta Llorell Linares, and I'm involved on the Permit COE project on behalf of MBLEBI, and I will be hosting this webinar today. Before we go any further in the webinar, I'd like to let you know that the webinar is being recorded and will be disseminated afterwards on the Permit COE website and our YouTube channel. After the presentation today, we will have some time to address questions from the attendees, so you can write your questions on the Q&A function in Zoom. And all the material that we share are licensed under a CC BY 4.0 license, except where further licensing details are provided in the material. Permit COE is the HPC Exascale Center of Excellence for Personalized Medicine in Europe. It focuses on simulations of cellular mechanistic models, which are essential to translate omics data into medical action. The performance of the current simulation software is still not enough to tackle certain medical problems, such as tumor evolution or patient-specific treatments. So Permit COE will scale up cell simulation software to HPC exascale system to enable the creation of models of cellular functions of medical relevance. And it will achieve this goal through a series of objectives. First of all, it will optimize selected cell-level simulation software to run in pre-exascale platforms. Second, Permit COE is working on a series of use cases to show the use of Permit COE solutions in areas of clinical relevance, such as tumor evolution or drug synergies for cancer treatment. Permit COE will also train biomedical professionals in the use of Permit COE tools. It will integrate the personalized medicine communities into the European HPC exascale ecosystem, and it will build the basis for the sustainability of the Center of Excellence. Today's presenter is Dr. Joaquín Dopazo, the director of the computational medicine platform of Fundación Progreso y Salud. He holds a degree in chemistry and a PhD in biological sciences. He has previously worked in public centers like the Research Center Príncipe Felipe in Valencia and the Spanish National Cancer Research Center in Madrid, and companies like Glaxo Wellcome SA. His field of work is personalized medicine with an approach that includes systems biology, mathematical modeling, and artificial intelligence. So, Joaquin, the floor is yours.
Firstly, let me thank the organizers for inviting me to share some of the ideas of this conference, which has a very long title. Actually, the title was going to be shorter, but after several discussions with some friends, they convinced me that, that what we were doing with the models was actually digital twins. I have always been a bit skeptic with the concept of digital twin, especially in medicine. I will comment why. But to some extent, something which is important and probably is the core of the presentation is the use of models or the use of approaches that at some point convey causality that can be used for prediction. So So that would be the definition of the digital twin. There are replicas of objects or physical systems that typically are used for simulating the events or intervention in the real world. Point, as I mentioned briefly before, is that in the context of medicine, digital twins have been proposed as a methodology, like in the industry or in other fields, but it's a quite controversial concept in medicine, given that build up such models or such digital twins requires an extensive knowledge of the system, and we are far away of having this detail of knowledge of the human body. That is at the end what we want to simulate. So I mean, that would be the idea. If we have a system, we can decompose the system in their pieces. We have different levels of organization within this system. For example, we have these pieces are that you have on the right. I don't know if you can see the mouse. This small collection of pieces would belong to a organ of the car, that would be in this particular case the engine, and we can simulate the way in which these things or these levels of organization work. So one thing are the pieces, and you have below the pieces at work. So if we can simulate the operations of the pieces, how pieces interact among them, and simulate this operation, you can simulate what this, I mean, the functionality of the system. In that case, most of the way in which the system works can be simulated, because we know absolutely everything. If we are in the context of medicine, we have a system which is immensely more complex than a car. And a car is complex, it's just a car, but this is much more complex and is poorly known. So we still lack a lot of knowledge. We have also these different levels of organization. And typically we think in the full body, we think of the organs, and we think of parts of organs, and we have at the end, behind everything, the molecular level detail. That would be this representation. And here the problem starts. So we don't have a clear idea of how these pieces work, how these pieces, in some cases we have some description of how pieces interact among them, but we don't know exactly how do they work. This modeling would correspond to a digital twin. So we cannot simulate, I mean, we are really, I mean, probably decades, or let's talk about decades, being optimistic, but many decades away from having a proper simulation of the human body, but still we can simulate some parts of the system. But as I mentioned, we neither know all the pieces, nor we know how to relate to each other. So then how realistic can our digital twin be? So what can we do? Obviously, it depends very much on the speciality of the people working with this concept of digital twins can simulate one or another level. But we typically want to focus the problem from the lower level and from the molecular level. So the idea would be we can have a molecular map that describe how molecules interact among them, proteins typically. We can model this interaction, and we can go from this molecular behavior to the behavior of the organ. I mean, that would be the general operation of the system. We can put some context when this general operation happens in a specific organ, or happens in a specific condition, in a disease or whatever, and then we go to the body. Obviously, we are not going to simulate how the body works. We are going to simulate specific phenotypes. So if we would be very happy if we could go from the molecular level to define a specific phenotype in the body. Then we use these maps, these molecular maps that are pathways. Probably you are very familiar with pathways. They are quite common. So you have many different repositories. Probably the most known is CARE. You have Reactome in the EBI. There is this wiki pathways. There are more. There are other more specific, etc. There are also initiatives like disease maps, in which they take specifically the parts of these maps that are relevant for diseases. So there is information, there is reasonable amount of information on the detailed map of cell activity that describe the way in which molecules interact to each other. But what is important here is not that the molecules interact to each other, is that why they interact. They interact because they finally ultimately trigger cell functions which are responsible for the behavior of the cell, and at the end the fate of the cell, but at the end the fate of the organs, at the end the fate of the patient, right? And this is what we want to simulate. So firstly, we have to focus on the concept which is important here, is the function. So we have these maps, but not all the proteins do things by themselves. So some of these proteins simply are transmitting a signal or doing, and the proteins typically do several things. So something which is important is to identify the level at which this map is describing an activity, a cell activity or a cell functionality, which at the end is a holistic concept. So it's not, we have to forget the protein-centric or the gene-centric interpretation and go more for a holistic interpretation. There are like 3 levels. One level would be the pathway level. I mean, well, let's start by the gene level. So the gene level is what we are used to. So we have this gene which is overexpressed, then we start to think why this gene is overexpressed and what would happen if I inhibit this gene or whatever. But the effect of a gene within this context is quite complex many times. For example, in this case, this is the summary. It's not because it's more complex, but it's a summary of the apoptosis pathway. And typically the pathways recapitulate biological knowledge on many things, and typically there are many functions that are carried out by the pathways, and typically these functions are contradictory, are opposite, not contradictory, but opposite. For example, this pathway, the apoptosis pathway, is responsible both for cell survival and for cell death, right? So if you just think of a specific gene, probably you will have not a clear idea of what is going on. For example, this gene BAD marked here, depending on the partners, it could be involved in apoptosis or it could be involved in survival. So having this gene active says nothing on what is going on in the cell. If you go at the level of the pathway, say, okay, there are many genes in this pathway which are active. So, well, but what is the pathway is doing? Is killing the cell or is making the cell to survive? So the point here is to go to the specific circuits, to try to look for the activity of specific circuits that at the end do something. So the first thing that we have to do is to identify this part of the pathways which are responsible for a specific function. And from a mathematical point of view, you have to identify those proteins. I mean, this is a directed tree, right? So you have arrows, and these arrows mean some type of interaction. That can be an activation, many different types of activation, or can be an inhibition. So there are arrows that go from one gene to other. So when you have one gene that receives an arrow, but there is not an arrow from this gene to other and has an annotation, this is the gene which is at the end of the cascade and do something, right? So we need to detect all the cascade and try to see if this cascade is active, so as this last gene can do whatever this gene is annotated to do, right? So the first thing that we have to do is to define these circuits, which has to do with functionality. So what we do then is to take a pathway and to identify these these effector nodes and decompose the pathway in all the pieces of the subcircuit that all the part of the subpathways that define this circuit. So at the end, you may have one or several receptors that finally downstream goes to a unique effector. So these circuits are our units of functionality, right? In which we are going to focus. So we use a specific type of mathematical modeling, which is called mechanistic, because typically it conveys causality, right? So the idea is that by using this model, we can provide a quantitative representation of what the cell is doing, right? We use pathways to define these circuits, also how proteins interact to each other until ultimately one of these proteins trigger a cell functional activity. We define these functional endpoints. And what is interesting here is that we use gene expression as data. And this is the main reason for which we are very, very focused on this molecular level. Because gene expression, nowadays, we have lots of gene expression data. I mean, really lots of this data. There are data which are cheap to obtain. They are quite accurate with the current technologies. And you have measurement for, absolutely anything that you can imagine. So there are this data really galore. So how one of these models work? Let's imagine that we have this circuit here on the top right, which would be a very simple circuit composed by two receptors, which would be proteins A and B. Then you have the rest of proteins C, D, E, F that has this topology, this specific topology. We have a different topology. And finally, if the signal arrives to G, then G triggers some function, typically by many ways can be done, right? Then what we can do is to infer what would be the activity, because these are proteins. So we use gene expression as proxies of protein presence, which at the end is a proxy of protein activity. Many people discuss that, you have a gene expressing, it doesn't mean that you have the protein. Yeah, good, but we are not focusing on a specific unique protein out of context. We are talking about the expression of a chain of protein which are really a biological subsystem. So if all these proteins all these genes are up, it's probably because there is a biological program behind that has accounted for this, right? So it's quite realistic, this assumption. So let's imagine that you have this condition A, in which we have active the genes A, C, D, E, and G. So you would put these genes over this circuit. You see that if A receives signal, the signal can be transmitted until G that does the function, whatever it is. But you can have a different condition like B, in which you have genes B, C, E, and G active. So you have, under this condition, if B receives a signal, the signal cannot pass because V is making a short circuit. So in that simple way, we use a quantitative formula. It's a little bit more complex, but we use this formula. So using the levels of gene activity, we can guess what is the level of activity for this specific function. So this is quite interesting because we can convert something that we can measure, which is gene expression, and is easy to measure and is cheap and is accurate, in something which is more subtle, which is a cell functionality. How can we measure cell division, for example, the activity of cell division? We should prepare a very complex experiment to measure, well, maybe not that complex, but other function can be really complex to measure. And that way you can measure the signal that the cell dedicates to trigger this function, which in theory should be related to the function, right? So sounds good, but does it work? Let's see. So we took the data of the TCGA, or the Cancer Atlas, or the Cancer Genomic Project, etc. And there we have a lot of data on gene expression of the tumors, biopsies, and we know what happened with the patient, if the patient dies or goes well or whatever. Then we can define functions that has to do with cancer. Probably you are familiar with this hallmarks of cancer, which were defined in 2011, and now they go for the third or the fourth definition. There are specific cell functionalities which has to do with the conversion of a cell into a malignant cell, right? So you can identify easily the circuits that trigger DNA replication. So you take patients. Some of these patients survive, some of these patients die. You calculate what is the level of DNA replication in the tumor of the patient, and what you can see here is that clearly patients with a high level of DNA replication die more than the others who has not so high the level of replication. Okay, maybe just by chance, but you identify other hallmarks like anti-apoptosis. So patient here on top right, patient with a high anti-apoptotic activity die more. They have a worse prognostic. Cells, you go top right down, patient with a low in that case, cell adhesion activity, which probably means that the cells go and float in the blood and can colonize and make metastases, distant metastases. So these patients die more. So they have a worse prognostic. For example, angiogenesis patient which have high angiogenic activity, they have a worse prognostic. So what we did here was to convert by means of the model gene expression into cell activities. And since we know what cell activities are relevant for the cancer, we can identify them, and you see that actually patients with these activities, with these cancer activities, they have a bad prognostic. Obviously, other unrelated activities has nothing to do with survival of patient, and they don't separate patient. And something that you have to keep in mind is that the activity, we are not measuring this activity. The activity is a construct. We have inferred the activity from the model. We are not measuring the activity. So we have a tool that to some extent allows us to convert gene expression, which is something that we can measure, into cell activities, right? So that's nice. That's nice. But there is a nicer property of these models. These models allow to make causal predictions, meaning that I can take a condition, for example, this condition here, that is represented here, where you have A, C, D, E, F, and G active. So the signal goes through with no problem, and the function is active. And then I can simulate, for example, knockout. I can simulate interventions. I can simulate knockout, but I can simulate overexpression. I could simulate the overexpression of B. I can just change the values of the condition and recalculate and compare and say, what would happen if I do something? This is a qualitative change, because I don't need to measure things. I can have a condition and say, what would happen if I would do that? For example, if I do a knockout on the E gene, probably nothing happens because the signal still passes through F. Maybe it's not that efficient, but it still passes through F. But what would happen if I kill D? The signal cannot pass through this point. So I have switched off the circuit. In that case, it's quite obvious, but you imagine the very complex pathway that we have. You can do that, and you can search for a specific intervention that have a specific result that you are looking for. So let me show you this. Again, it sounds good, but it's realistic. Yes, it is. Obviously, it's a prediction. We cannot pretend that all the prediction work, but it works quite well. And actually, this is probably the best paper of my career. This is the paper from which I'm really proud of this paper. So what we did here was to predict a knockout that was never done. Well, I mean, we predict several, looking for a knockout that kills or reduce the viability of one cell. And we went to an experimental group and we did with them the validation. And actually, the predictions were correct. So you see this type of approach is completely different to the approach that we are used to. Typically, these type of things are driven by experimental people. And sometimes people who just analyze data, they say, okay, can you analyze the data? Yeah, and you do the analysis. But the ideas come from the experimental part. So now we are in a moment in which we can start to think in a different way and say, okay, we have model, we have data, and we can start to formulate hypotheses from the in silico directly, right? So this is an introduction to this type of models and to show you that they work. So why repurposing? Why we are interested in repurposing? I'm going to talk to you about repurposing in emerging diseases, and if we have time, I will tell you another field in which repurposing is also interesting, which is rare diseases. So in the case of emerging diseases, and this is the case of the COVID pandemics that we have a few years ago, and we still have. And actually, we have now two strains which are very very contagious. So be careful, because now the number of people that goes to the hospital is increasing. The number of hospitalization is increasing. So what do we need in one of these situations? We need something quick. We need a quick response. Quick response, you cannot start from the scratch with a drug. So something which is interesting is, let's see if we can repurpose any type of drugs. We need a quick implementation. A drug, because this drug is already in use, can be quickly implemented because it has a known security profile. it has a known mechanism of action, is already registered. So legally is quite easy in comparison with a drug from the very beginning. What is the problem with emerging disease? But most of the details of the real mechanism of the disease are unknown. So we have to try to see how can we relate this the drugs and the mechanism. So what would be the solution? The solution would be to use this method that I told you, that I have been commenting. And how can we solve relationships that are beyond the models using causal machine learning? So I'm going to talk about that. So in this moment of alert, so there were different projects. There was a lot of money. There was money. Never has been a lot of money in this country for research, but then there was money, and they funded several projects. So we got this project, and we took advantage that we were involved in this large-scale project, in which they were defining the COVID-19 disease map. So there was a large collaboration that involved more than 230 different biocurators, domain experts, modelers, data analysis, etc., from 120 different institutions in 30 countries. So it was a quite interesting initiative. So finally, they come up with a quite detailed map that we can use as basis for the modeling, right? So we modeled it and we published it in a couple of papers, etc. But here is the important. So we have the map, right? So now we can model the map with the algorithm that I mentioned. The problem is that, okay, if other drugs have a target in the disease map, so the effect of this target, of the effect of the drug on this target, can be easily modeled, as I showed you before. So we can just put the expression data and say, what would happen if I inhibit that? Oh, that's good. It will inhibit this part that has to do with respiratory tract or has to do with coagulopathies or whatever. But what happened if the drugs are outside of the map, which in many cases is what happened. So this is a problem. This is a problem because we cannot model that. I mean, we cannot directly model that using the mechanistic model, right? Then we have to imagine how to relate that with the map. So at the end, the map, as I mentioned in the very beginning, is a collection of proteins and arrows that link these proteins. Any of these arrow is biological knowledge, is a lot of experiments and validation, etc., demonstrate that this protein actually is activating or inhibiting this other protein and no other, only this protein. So then you draw an arrow. And this is a lot of experiments, etc., right? And the problem is that currently 1/3 of the genome can be modeled. The other 2/3 are not linked are not part of this relationship, right? In other words, we don't have a real knowledge of what is going on between these proteins, how these proteins interact to do something. This is one of the reasons for which this digital twins has a lot of limitation in the field of when we want to make a real digital twin, right? And the problem is that drawing one of these arrows, it takes a lot of time. It requires the generation of this biological knowledge, and it takes time. So maybe we don't need to do experiment because we have lots of experiments. We have lots of gene expression data, and maybe we could use this to in the context of machine learning to try to learn biology from the data. So I mean, the idea is fantastic. It's okay. Why not? I mean, in other cases, you can learn. For example, there are lots of applications, machine learning and deep learning, etc., which they can learn, for example, in image. In the case of image, it works very well. So you can take radiographies, or you can take some different types of medical image, and you can decide if there is a lesion, or there is a problem, or whatever. And in some cases, the level of accuracy is even higher than the human experts, right? So at the end, the software, the network, or the algorithm, the machine learning algorithm is learning from the data. And you mark the data, you say, okay, you can learn what happened with these pixels, or what pixels are important, and then you learn how to identify something. So this is something that we could do in theory. We could take all the gene expression data and say, okay, just learn how proteins interact among them, and what type of interaction is. Well, I mean, it is realistic. No, it is not realistic, because no matter that we have lots of data, we have the course of the dimensionality. So we have still more variables, and the interaction of the variables are much more complex than the interaction that we have pixels in an image. That pixels at the end, they are important pixels around, not the other pixel in the other corner of the image. So in principle, we are not going to solve all the biology in one shot. This is for sure. But something that we can do is to reduce the dimensionality of the problem, and not being so ambitious, not trying to learn all the biology in one shot. But something that we could do is, okay, we have a part of the biology that we know, that are these circuits, and these circuits involved in the disease. And then we can try to see, okay, what are the relationships not between all the proteins and all proteins among all the proteins, but I'm interested in a specific set of proteins, which are these proteins that are targets of drugs already in use. And I can try to learn what are the relationships between these proteins, which are targets of other drugs, and the proteins which are in my map of the disease, right? And in that case, is something which is affordable, the problem. So what we did was to try to learn. Well, I mean, the idea is we try to predict from the activities of the targets, of different targets. We try to predict the activities of the circuits, right? Obviously, most of the drug targets cannot predict anything, because they are completely unrelated, but some of the targets can predict some parts of the activity of this map, right? We use here, I say, the multi-output random forest regression that was used in the original paper, but we have tried other approaches. They work more or less. I mean, there are 2 or 3 approaches that can work. And something which is more important, that use this SHAP score, which are used to look for the relevance of any of the targets, because obviously there is always an interaction, but what interaction is relevant and what interaction is not relevant. So in that way, we can use this multi-input, multi-output, because the inputs are all the drug targets, and the output are all the circuits, and say, okay, can we predict one for the other, and what is important in predicting, and what, right? So what we have are diagrams like this, in which we can see that some specific target, but the rest are not important, as you can see here, but some specific target were important for some specific, these are the drugs on the target, for some specific or general parts of the pathway, right? Then we are not interested in those drugs which are acting on absolutely everything, because I mean, probably they're killing the cell and that's it. And actually, it's curious because you have the chloroquine that was very famous at the very beginning, because they say, oh, chloroquine works. Yeah, the chloroquine works and kill the virus, kill the cell, kill you, and kill everything. So it's not what we are looking for. But there are other more specific drugs which are more specific of some hallmarks of the disease. So they go directly against, for example, against the immune response, exacerbating immune response, against energetics, inflammatory response, etc., etc. So when we start with this, we made a prediction and said, okay, we didn't have any means of checking if they are real or not. So something which was nice was to realize that in the moment in which we were publishing it, they published this paper. Was a review on the trials that were testing treatments for COVID-19, and there were trials for all the. I mean, we have predicted targets for all the drugs that we're trying here that have a target. Obviously, other drugs didn't have. Other treatment didn't have a target. Some of them were something like inhalation of gas or something. They don't have a specific target. But those who have a target were predicted by the method. So you say that's okay. No, it's not bad. It's nothing sure, but we are at least oriented. So what we thought is, okay, let's go for real data. So we have this big database in Andalusia, which is the population database, and all the health system, which is the device here, as in other places, all the system every month on monthly basis, they dump all the information on this database, which is consolidated. So it has information of absolutely everything that the system has recorded on a patient for over 15 million patients of the health system. So what we did was, given that there were lots of problems for using the data, what we did was to build up a trusted research environment, which is essentially a computational place in which we can do all the calculations that you need, but in a secure and controlled place. So what we did was to set up this infrastructure, which is available for research, which is a computational infrastructure which resides within the network of the hospital network, so it's not outside. So in that case, it's quite easy because you can request. Now that would be the typical. On the right is the typical pathway that follow with the typical analysis of clinical data. Typically, you request the data to this committee, and there is an approval. Then they allow you to take the data. The data must be anonymized. They are extracted and they are used. So in many cases there are problems, and the ethics committee reject the project just because there are doubts that of security outside. So what we did was just internalize that, so the analysis can be done within the hospital in a trusted environment, and the only thing that is liberated is the result. So in that case, it's quite nice. So, okay. Then we start to check some of the drugs that we predicted. We started with this because there was a lot of interest in the community, because there were several groups working on divitamine, and actually there is the calciferiol or vitamin D has a clear protective effect, and I'm still having this calciferiol because it protects. It is a factor, right? That you can see here. It has a significant protective effect. And this is not just that you see what happened with the patient that have a consume of vitamin D or not. Is that you take into account all of these variables that could be affecting, which are sex, age, other vaccination, other comorbidities like diabetes, disease, cancer, etc. And you have to take into account that, meaning that when we say that something is protected at any age, at any sex, at any comorbidity, at any etc. Right? So it's not that there is a correlation; it's a causal analysis, right? So we did the analysis in more detail, and we came up with 21. This is in second revision, so probably will be published soon. We come up with 21 quite effective treatments. Some of them are really effective, right? And one, I mean, actually there are more, but they are not significant because there were few people. But some of the drugs were counterproductive. They increase the mortality. What is interesting is that we can not only see if it has an effect, a significant effect in the mortality of the patient. We can see also if there is a biochemical background behind. So we have access to the analytics, and you can see how the lymphocyte count in these cases, in the cases of the drug that protect, actually goes very well. While, for example, in the case of the drug which is causing more mortality, you have a lower lymphocyte count. So there is a biochemistry behind which is validating this. So what is interesting is that there is an enrichment. So many of these drugs were predicted by us. So this is something that at the end makes us very happy, because at the end we were really predicting correctly. So we did the prediction, and then we could do the validation using this real world data. I was going to mention something on the rare diseases, but I'm going to jump a little bit because maybe we are short of time. But I mean, it's a similar case. So we essentially did other molecular mass we make in an industrial way many molecular mass for many rare diseases. And then we are validating now the results. So we have some cases which they have validated some of the cases with this previous publication. Now we are publishing that for retinitis, for anemia, for anemia, for others. So just to wrap up a little bit, I think the take-home message is that what we did essentially was to model biological knowledge. So to formulate the hypothesis, to learn how these proteins relate to the maps using public data, and to do a validation using real world data. All the process from the formulation of the hypothesis to the validation has been done without doing any single experiment. Of course, this experiment were already done. I mean that we particularly didn't do any single experiment. But the important message is that there are lots of data. So for many things that you can do, there are already data that can be used. This is, to me, this is a new paradigm for knowledge generation, because at the end you can use modeling. You can do modeling which involves biological knowledge and is causal, and you can try to make prediction, and you can validate prediction using real world data. This, I mean, is far away from what we would expect for a digital twin, but it's a little twin, the little digital twin that actually works. So finishing here, and I'm really happy. This is the group. These are our sponsors, etc., etc. This is the hospital where we work. It's Virgen del Rocío. And I'll be happy of taking questions you have. Thank you.
Thank you very much, Joaquin. Yeah, as Joaquin said, if you have any questions, you can write them in the Q&A function in Zoom. And while you do that, I can ask with a general one, Joaquin. So you're telling us now you did all this without experiments that you did, because the experiments on the data were already there. How do you see that going forward? So the relationship between the models and the experiments, will you have like a circuit where, you know, models inform experiments and then inform models, the experiments inform models and all that? How do you see it?
Well, I mean, the way we see that is that you could enrich the map that you have by validating that your prediction, for example, you predict that drug is what happened here. You predict that drug has an influence on this circuit, and it will prevent coagulopathies. And actually, the patient has no coagulopathies. This is what I observe in my patient. Then I can draw this new line. So for my next simulation, I have a new arrow. So I have a more complete map, etc. So at the end, this is a raw way of learning biology from the data by try and error.
Yes. Thank you. So there are a couple of questions now there. So one is how to distinguish a correlate relationship from a causal relationship.
Yeah, good question. And this is the core of the presentation. So, fairly, the model is causal. So the model is causal by principle, because we already know that, because there is knowledge that we already have, that this protein activates the other protein and activates the protein, and the final protein is a transcription factor that transcribes something that does something, right? And in that case, if I do something here, there is a consequence here. So the causality is built up in the model. A different thing is, how do you distinguish in the case of this influence that we are used with the machine learning model? How do you distinguish between correlation and causality? This is not formal mathematical causality, but we are using models, and we are using the sharp information, which are something which looks very much causality, right? So they're not mathematically formally causality, but there are machine learning models that involve causality. So it's not just a correlation. It's that when this is up, okay, but this might be up. So it sort of checks, since it's multi, I don't know how to explain easily, but since it's multi-input, multi-output, it removes the possibility that there are other different ways, and can be just a correlation, and the cause could be a different one. So the short answer is that we use machine learning models that, to some extent, consider causality. It's not just a correlation.
Thank you, Joaquin. So next question is, what number of minimum studies are needed as prior information to make a decent digital twin and prediction?
I don't know how to answer this, because, I mean, by definition, what we are doing is not exactly a digital twin. So for doing a prediction, for doing one of these predictions, it depends very much. When we do a prediction of what would happen in this condition that I mentioned at the beginning. So we have the model, and you can simulate. You have a condition. You have these genes up, these genes down, and you can say, okay, what would happen if I remove the gene, or if I put the gene inactive? This is a comparison one-to-one, and there is no test behind. So it's simply to detect where, because I show you very simple circuits to illustrate that, but there are much more complex. So what we do is to say, okay, what would happen if I make an intervention here, and then you have some circuits which are with change, right? So it's not, when we are within the model, we are not making any type of test. We are just pointing out the places in which the system changes, and then we go for a different type of confirmation. That could be experimental, or it could be other type of confirmation. If you are more thinking of, for example, of the cases of the survival, I say, okay, is this function involved in survival or not? Then it depends very much on the case, because depends very much, because in some cases, what happened in biology and maybe in medicine, that in many cases you don't have the number of patients that you like, and then when you draw the confidence interval, you say, okay, this patient died more, but it's not significant because the confidence intervals overlap. And then it's very, very, again, a short answer is depends very much on what you want to test exactly, like in many other scenarios.
Okay, thank you, Joaquin. So I'm going to share my screen with some announcements. We'll stay, if Joaquin has the time, to answer all the questions, so you can see them on the video afterwards if you need to run to another meeting or so. So our next webinar will be next month on the 5th of October, and we'll have 3 of the participants of the summer school who won the poster prizes, and will be presenting the research on cancer, tailored cancer therapies, high-grade serous ovarian cancer, and communication in the tumor microenvironment. This will be 1 month. If you want more information and to register, go to the PERMET COE website. Then I also have an announcement on an MBORD workshop on computational models of life from molecular biology to digital twins. So more on digital twins. This is going to be at the end of November, close to Barcelona, northeast of Barcelona, and you still have time to send an abstract before the 18th of September, and then the registration is on the 16th. So now, Joaquin, we continue with the questions. So there's a comment there. This is very interesting. Thank you for all this work. Congratulations to the team. How do you see this being adapted to a multi-omics data set of the same set of participants?
Yeah, actually, I have not mentioned that, but we have other multi-omics approach for this, and we have several papers published on that. So we look for activity at the end, right? So we think that you have gene expression plus mutation. You recapitulate a lot of other omics, right? For example, you may have a copy number, and you have the gene over a lot of copies, and then you will have higher expression. Then what we do is to combine the expression plus the mutations, which is the activity of the gene and the functionality of the gene. So a gene can be active, but can have, for example, a frameshift mutation, so the protein is not active. Then we remove from the model the protein. How can you extrapolate that if you don't have this information? For example, you have, instead of having gene expression, you have methylation. you may, I mean, something that we do sometimes is to say, okay, that the part in which this methylation will affect, we take the normal situation and we go the gene down or the gene up, depending on the level of methylation. I mean, you can do some tricks in which you can reflect this multiomic data in terms of gene expression and gene integrity, right? So at the end, what we play with these two concepts. Obviously, there are. I mean, this is just to see where does the change affect. We cannot be sure that this change, the degree of affectation is real, but it's something that we do.
Thank you. So we'll go to the next one. Can you please give details on what is considered a circuit? How is the line drawn between which nodes are part of a circuit and which nodes are not, considering that nodes may have a large amount of edges?
Yeah, okay. This is, I mean, this is if you imagine, you visualize, for example, signaling circuit, you have proteins, and the proteins are connected by arrows, right? The arrows are directional. It's not like, I mean, this is for signaling circuit. It's not for metabolism. Metabolism, the arrows are not directional. You can go up in one direction or the other direction. Now we have the signal traveling in one direction only. So you have these arrows. Then you have proteins which has an arrow pointing to them and no arrow. And typically these proteins are annotated with a function. So these are the effector proteins. And then what we do is to find all the effector proteins that do things in the circuit and then go backwards, no matter how complex it is, until we found a protein which has an arrow to other but has not an arrow to itself, which is the primary receptor. So we travel from the effector to the receptor, or in many cases there are several receptors. There are many receptors, and it can be as complex as possible. But at the end, what you do is like taking the end of the circuit, say, okay, you go backwards, and what happens is that many of the circuits share protein with other circuits, and this is completely normal. And many times, what happens is that you say, okay, when you see the activity of the genes in the pathway, it makes no sense. Then if you individualize circuits, you say, okay, now it makes sense because you have enlightened only parts of this pathway which correspond to a circuit. And this is what we do. So we go from the effector backwards until the receptors. In some cases, the receptor, all the receptor, that's it. Okay, it's one big circuit. But in many cases, there are like, I show a slide in which there were decomposed. So at the end, what do you remember one sort of animation?
You can show it if you want.
Yeah, let me go for it. It's at the beginning. I think this one. Can you see it? This is a very small pathway, but what you can see is these proteins, and this is square. This is the function annotated. So all these proteins which are a square, this square is not part of the circuit. This square is annotated by us, and this is the function. So all these proteins, which has this square, which is the label, are final proteins, right? Are effectors. And then what we do is to get, okay, this is an effector, and you go from the effector backwards until the well, I mean, here exactly. until the receptors. You see here, can you see the mouse?
Yes, we see the mouse.
Okay. So you see, okay, what happened with this protein? Because it's connected here, but you can see that this protein is connected not from here to here, but from here to here. So what we do is, okay, arrows that point here, that arrows that point this, and this, arrows that point here, here, and here. Arrows that point this one, no arrows. There are no other protein pointing here. So for this circuit, if the signal is going here, it goes there to this function. Obviously, the function, it can go through this way, but this is a different circuit. So at the end, mathematically, it's very easy. You only look for, you go to the last protein and you look for things that are pointing to you until you find that somebody who is not pointing to anybody, and that this is the circuit.
Thank you very much for that detailed explanation. So we have a few more questions. So there's another, thanks for sharing. What would you say to people that believe that there are many variables that we are not taking into account in this type of modeling and research, that the puzzle is not completed until we integrate the extra layers, for example, the extracellular matrix?
Well, I mean, what I have been saying all the time, that this is not perfect. This is what we can do in a very simple way, and this is the little digital twin. There is a lot of detail that is not here. This is true. And I tell you that in cancer, for example, this type of simulation works very well because many of the things that happen in cancer is a matter of signaling. So in that case, it works very well. For example, in rare diseases, and there are many rare diseases that has nothing to do with signaling, and then we have a lot of difficulties to make the simulation. So as you say, the puzzle is not complete, and we can say only a part of the reality. As soon as we have more arrows and more relationship, and we can model more relationship, we will have more reliable digital twins.
A second, because Vivian, who is the one who asked that question, raised the hand. Vivian, do you want to talk?
Yes, can you listen to me?
Yes.
Yes.
Yeah, great. Yeah, I asked the question because I have faced this challenge with experimental people, and they're not very convinced that the modeling that we are doing is on the right track, or we're asking the right questions. They feel like we are taking too broad considerations, and I just ask you as an expert, what would be the advice how to deal with this? Because I believe in this, but many do not.
Yeah, well, but this is, I mean, we have found this problem many times. So
Yeah.
experimentalists, I mean, when you invade the ecosystem of other people, so they are very protective. So many experimental people don't believe in that mainly because they don't understand, firstly, and secondly because they feel that if you can do with a simulation what they do with months of experimentation is something which is a bit depressing for them, right? So, I mean, there is evidence and there are papers that demonstrate that works. We have published a number, not a lot, but maybe more than 10 papers, 15 papers, and some of these papers have validation. So, to me, it's a demonstration that works with limitations. And typically, what I would recommend you is to try to use some type of persuasion and reverse psychology in a way where they feel that they are at the end they are going to give you the link that lacks here or something like this. But it's more a problem of self-confidence of experimentalists than a real problem, because this model, I mean, with the limitation that they have a lot of limitations. But for example, in cancer, they work fantastically well because, I mean, they predict many things, but very, very accurately, because cancer is mainly a disease of signaling, right? Then, I mean, but it's just collect evidences or something like this.
Yeah. Thank you.
Okay, thank you. So we have another thanks for the talk, very interesting. So according to you, what is the proportion of edges in biological networks that relate to indirect and real direct relationships? And how do you think these affect the predictions of your models?
So, what is this?
What is the proportion, is Jean-Baptiste Mourlon, what is the proportion of edges in biological networks that relate to indirect and real direct relationships? And how do you think this affects the predictions of your models?
Direct and real direct. You mean that they are real or not? Or you mean how do we trust in these edges? Or you mean that I would say that in the, I mean, something which is very easy is when you compare different.
Wait a second, because Jean-Baptiste have raised the hand, so maybe he can clarify.
Hi, can you hear me?
Yes.
Yes.
Awesome. Yeah, the question is related to when we're doing like a causal inference, or we have some causal relationships, we can have like causal relationships, but these relationships are not physical. You know, we just know that one activity is like the appearance of others of the activity of downstream different entities, but it's not directly related. There's no physical relationships between the two. So the question is, do we know today what is the proportion of real, let's say, physical relationships and the relationships that are causal but not, let's say, physical? I don't know if it makes sense.
Yes. We take, I mean, we use mainly for this, we use KEGG. In the case of KEGG database, there is a quite clear relationship between, quite clear description of the relationship between the proteins. And we separate in two classes, activation and inhibition. And the relation is, I mean, there are lists, phosphorylation, ubiquitination, etc., etc., etc. Some of them are physical interaction, and some of them maybe they're not physical interaction, but at the end they phosphorylate or whatever. So I don't care if they are physical or not. I only care if the type of interaction, if the interaction is an activation, so it's transmitting the signal, or is not transmitting the signal. How reliable are they? When you compare different databases, there is a core which is constant among them. So probably these are the more reliable. And in signaling, there is a very large part of the signaling which is probably is quite solid in terms of reliability. So you can trust very much this. There are other, I mean, you go to Reactome or you go to other databases or to wiki pathways. So there are some suggested relationships that maybe they're not real. So probably there will be a process of curation in time. But I think that you can trust in what is in the core of the signaling. You can trust most of the relationship, and you can trust that they are causal, no matter that if they are physical or not. This is my opinion. The number, I don't know.
Okay, so thank you very much. I hope, Jean-Baptiste, that answers the question.
Yes, thank you.
Then
Thank you.
go for the last two ones, and then they will all be in the recording. So we have one more comment about thanks for the nice talk. And this is a bit of a technical question. Why SHAP in particular for the machine learning model? Did you try any other explainability approaches?
I will tell you. We use SHAP because it's the favorite of my postdoc who was doing that. We are planning to use other causal approaches, but we have used that. Actually, the most popular as well. So we wanted to go for something over which there is a consensus. Many people use that, but there is no vision beyond that.
Okay, thank you. And then the last one is, thanks for the interesting seminar. What would be the differences in applying this modeling in studies with bacterial species as variables, not genes? Would that be possible and equally promising? What should have to be changed?
It could be done. I mean, simply you should have a different map, which would be the signaling within the bacteria, which is far simpler than the human signaling. And it is possible. Actually, not on the signaling side, but on the metabolism side, there are lots of studies with models of metabolism have been made in bacteria precisely because they are simpler. Something that you can have a look. You can have a look at the metabolic modeling in bacteria, and you will get a lot of publication.
But I guess this doesn't. As I read the question, and I know almost nothing about it, it's not about one bacteria and what happens in a bacteria. But if you have several bacterial species together, because it says using the bacterial species as variable instead of the genes. So I guess it's you have one bacteria here that interacts with this other bacteria and interact with that other bacteria.
I misunderstood. I mean, why not? But I mean, this model are for transmission of information. If to some extent this bacterial species do something that can be assimilated to transmission of information, meaning that this bacteria needs the other bacteria because otherwise it wouldn't survive, whatever, you can make some type of network among all the bacteria, all the different types of bacteria. Probably you could do something like this. Maybe it's not the best approach, but it would be interesting to think of that.
Okay. Thank you very much, Joaquin. Thank you very much to everybody who has stayed with us until now. And yeah, see you next time. The video will be available, as I said, and you'll receive an email about that. So thank you. Have a nice day. Bye.
Thank you. Bye.